Okay, so we talked about anatomy of the thyroid gland. We've talked about and remember it's this butterfly shape, left and right lobes. It's got these circular follicles with the colloid inside. It's got these C cells and blood vessels in between the follicle cells. And then we talked about synthesis of the thyroid hormone. We talked about the trapping of the iodine. And we talked about making of the thyroid globulin train, right? Different pieces from the tyrosyl groups, and how you make your MIT and DIT, and then cleave these and secrete the hormone. So I want to back up a bit and talk about how that process is triggered. Remember, we talked about thyroid stimulating hormone and thyroid stimulating hormone receptor. So I want to talk about how the thyroid works within what is called the HPT axis. So the hypothalamus pituitary thyroid axis. So that axis looks like this. You have secretion of thyrotropin releasing hormone, or TRH, by the hypothalamus. This then stimulates the anterior pituitary gland to secrete thyroid stimulating hormone that we talked about. This TSH then binds to the thyroid stimulating hormone receptor located within the thyroid, leading to that iodine trapping, and then eventual secretion of T4 and T3 by the thyroid gland. Now, one of the important things about T3 and T4 is they're majority protein bound in the plasma. And then just showing you, remember I talked about the ratios of T4 versus T3. So the majority of the thyroid gland secretion is in the form of T4. Now, the hypothalamus also contains thyroid hormone receptors, which can be detecting this negative feedback. So the thyroid gland, this is how you turn it on, but the thyroid gland itself, by releasing T3 and T4, can inhibit the hypothalamus. All right? That's that long loop feedback, remember, that we talked about. So thyroid hormone receptors within the hypothalamus will bind and detect low levels of T3 and T4 and then block release of this TRH. Now, assuming that there is no binding, right? If the T3 and T4 levels are very low, then you will see stimulation of the hypothalamus. Okay, so these receptors respond to whether or not there's high or low levels of T3 or T4 in the circulation. Low levels of T3 or T4 will drive TRH release. TRH will then stimulate the TRH receptors in the anterior pituitary and then secrete thyroid stimulating hormone. TSH is the major stimulus of thyroid gland and stimulates the trapping that we mentioned and eventual synthesis and secretion within the colloid. Now, how does the thyroid releasing hormone affect pituitary function? First, you have thyroid releasing hormone binding to a receptor within the pituitary. This drives phospholipase C activation, which will then eventually increase intracellular calcium concentrations, which then will cause both increased secretion of thyroid stimulating hormone from the anterior pituitary, and eventually drive synthesis and glycosylation of more hormone. Importantly, glycosylation is not necessary for the full biological activity of TSH. So TRH binding to the anterior pituitary drives both synthesis of the hormone and release of the hormone. Eventually, you can also get synthesis of TSH via increased PKC, which is that protein kinase C we mentioned earlier. Which also will drive TSH secretion. Now, what are the actions of TSH once it binds to TSH receptor within the thyroid? Remember, the first step was activation of the sodium iodine pump to uptake iodide and do that iodide trapping. It will then drive also synthesis and secretion of the thyroglobulin chain that we talked about, the long molecule that has cleavage. It also is responsible for the generation of hydrogen peroxide and iodination of tyrosine. And it drives secretion of T3 and T4. TSH will also influence blood flow through the thyroid. That's a key aspect for both bringing in more hormone and releasing T3 and T4. And importantly, in this last one, remember we mentioned this TPO enzyme. So TPO is thyroid peroxidase or thyroperoxidase enzyme, and expressed mainly in the thyroid, and it's secreted into the colloid upon TSH stimulation. TPO is responsible for that first step. Remember when we said we trapped iodide ions? They came across the membrane and we had to trap them there. So it converts iodine to form iodine and prepares that iodine to then be added onto the tyrosine residues. So TPO is a key enzyme for the initial steps of synthesis of thyroglobulin. And in humans, thyroperoxidase is encoded by something called the TPO gene. So it also is explicitly regulated. And finally, actually I wanted to say one other thing about just a summary of TSH effect. You can see that TSH has a dramatic effect on all steps of T3 and T4 production, synthesis, and release. Okay, so we've talked about the anatomy of the thyroid. We've talked about synthesis of thyroid hormones. We've talked about how they're regulated by anterior pituitary and TSH. And now let's talk a little bit about the mechanisms of action of thyroid hormone. So thyroid hormones, remember, are insoluble in water, and therefore they're transported in a blood-bound form into plasma proteins. Okay? So remember, I mentioned that 99% were bound to plasma protein. And if you think about the combination of T4 prohormone and the half-life, in addition to bound being protein, these are extremely protected hormones within the blood. The majority, about 75% of T4 binding, is to a protein called threonine binding globulin, or TBG, whereas 15 to 20% is bound to a different protein called trioxine binding prealbumin. And you can imagine that these proteins are also regulated, right, with another 5 to 10% bound to albumin. Only free unbound hormone is biologically active. So we got multiple steps here to achieve an active hormone. First, you have to cleave at the local tissue. You have to convert it from T4 to T3. Well, it's actually, first you have to release it from the protein that it's bound to, and then you have to cleave it from T4 to T3. Okay? So these molecules are extremely well protected, in part because they have such physiologic implications. Now, just a quick reminder, think about how hormones are different compared to the peptide hormones, what we talked about, or the steroid hormones, right? So this hormone is interesting because it comes from tyrosine, which is relatively soluble, right? Yet it's converted into something that's insoluble in water and acts like a cholesterol molecule. Okay, so why do we take such protected care of this molecule? Why do so many steps, so many controlled steps for synthesis? So important to control its reservoir, so important to control binding to the protein and releasing. Why is it so regulated? What does it actually do? So these are the mechanisms of action of thyroid hormones. So just a reminder, remember, we've got this TPG hormone bound to our in the blood. It's T4 bound to our blood plasma protein, right? And then in the case of T4, this is converted to T3, right? Via monodeiodinase, which can then, in the T3 form, bind to thyroid hormone receptor, which immediately can activate gene transcription inside the nucleus. Again, very similar to the cholesterol hormones. This will drive, in combination with retinoid receptor, drive gene transcription by binding to the thyroid response element, or TRE, leading to increased messenger RNA. And what are these messages? These messages, one, will drive sodium-potassium pump changes. It will drive gluconeogenic enzymes, which have to do with your metabolism. It will drive respiratory enzymes, again having to do with cell metabolism. It will drive myosin heavy chain, which can drive and influence trafficking. It will alter beta-adrenergic receptors, which have to do with a variety of things, including metabolism, heart rate, body temperature, amongst others. And then many other genes are also can be impacted by thyroid hormone. So, what is this retinoid X receptor complex that we're talking about here with the thyroid hormone? The thyroid hormone receptors regulate gene expression by binding to this hormone response element, this TRE, on DNA, and it does this by binding either as monomers or heterodimers with this retinoid X receptor. And this is then in turn activated by binding to 9-cis-retinoic acid. So you have an additional step here for regulation of this complex, which will modulate which genes get turned on when. Importantly, these TR-RXR heterodimers are the most transcriptionally active form of thyroid receptor transcription factor. Typically, you'll have TR-RXR binding here as a complex to this thyroid response element. Now, remember, we also said about 5% of thyroid is also made as T3 directly. It has only a half-life of 1 day. T3 in the blood is bound to TBG. And in this case, you don't need conversion of monodeiodinase. You can just allow the T3 that is released to immediately bind and enter into the cell and bind thyroid hormone receptor. Again, this is a less common route. The more common one is via the circulating T4 and conversion of T4. And why is that, remember? Because T4 is the more stable predominant form. Okay, so remember, the receptors for the thyroid hormones are intracellular, more specifically are nuclear. Remember that the thyroid hormones influence gene transcription and thus protein synthesis. And remember, we talked a lot about timing of receptors and timing of hormones and the differences between timing of hormones. And this is a good example where in the case of thyroid hormones, you see a 12 to 48 hour delay before any effect is observed, physiological effect is observed. So several weeks of hormone replacement therapy are required before deficiency in a patient is corrected. So if you measure thyroid deficiency in a patient, don't expect to see changes in a few hours. It's going to be several days or weeks of treatment before you will expect to see any changes manifested. So, again, I want you to think about comparing thyroid hormones versus cholesterol hormones versus peptide hormones. How are they stored? How stable are they? Are they protein bound or not protein bound? And for thyroid hormones, they are. Okay, almost all of them are bound to one of those three protein populations. Okay, so now let's talk a little bit about the biological effects of thyroid hormone. I mentioned to you all the messenger RNAs and proteins that are impacted, types of proteins that are impacted, and you can imagine that these could have some severe impact on physiology. So thyroid hormones are essential for normal growth and development. If you remove the thyroid, you can prevent metamorphosis, for example. So this is a desiccated thyroid was fed to a frog tadpoles, right, baby frogs, and this can induce metamorphosis. Whereas a hypophysectomy of a tadpole caused the thyroid to atrophy and prevent metamorphosis. So you can block this step by just removing the thyroid. They then did injection of pars distalis extracts, and this also induced metamorphosis of hypophysectomized tadpoles. So it promotes growth and it promotes development. It required for growth and development of the fetus. So in biology, molting, sloughing, or shedding in many invertebrates is the manner in which an animal routinely casts off part of its body, right? This can be snakes who cast off part of their skin, and it's often an outer layer or covering. So often some of the beetles do this, either at specific times of the year or specific points in the life cycle. And again, this is often driven by a thyroid hormone. And that's because thyroid hormone drives and controls the rate of growth of our tissue. Thyroid hormone also impacts on reproductive function and development and function of our central nervous system, in particular myelination, which is the protective part of myelin over our nerves. Thyroid hormone also impacts skeletal growth, okay? And then I mentioned to you in humans, it's often hair growth, but in reptiles and birds, it'll be molting of skin or feathers, okay? So you can see the snake here molting, and then you can see this is a bird changing feathers during the season, all driven and controlled by thyroid hormones. And then for amphibians, I mentioned to you the tadpoles. That was one of the early discoveries and studies of growth hormone and thyroid hormone, was around the metamorphosis of amphibia. Okay, so one of the main effects of thyroid hormones is to impact and increase the basal metabolic rate. And this is done by increasing the number and size of mitochondria and the activities of those important enzymes. So if you remember, we looked at the genes that were turned up or down, okay? And a lot of them had to do with mitochondrial function and metabolism. So thyroid hormones influence all major metabolic pathways. Their most obvious and well-known action is an increase in basal energy expenditure obtained by acting on protein, carbohydrate, and lipid metabolism. With specific regard to lipid metabolism, thyroid hormones affect synthesis, mobilization, and degradation of the lipids, although degradation is influenced more than synthesis. So the main and best known effects on lipid metabolism include enhanced utilization of lipid substrates, again in part by modifying those enzymes we talked about, increase in the synthesis and mobilization of triglycerides stored in fat, increase in the concentration of non-esterified fatty acids, and increase of lipoprotein lipase activity. While severe hypothyroidism is usually associated with an increased serum concentration of total cholesterol and atherogenic lipoproteins, the occurrence of acute myocardial infarction, heart attack in hypothyroid patients is not very frequent. So despite this increase in cholesterol, it doesn't always or usually lead to heart attack. However, hypothyroid patients appear to have an increased incidence of residual myocardial ischemia following a heart attack. Now, even in subclinical hypothyroidism, which we'll talk about the numbers, which is characterized by raised serum TSH levels with normal serum thyroid hormone concentrations, mild hyperlipidemia is present and can increase the risk of atherogenesis. So it is usually prudent to have a substitution therapy with L-thyrodoxine indicated in patients with both overt or subclinical hypothyroidism. And this is with patients with or without angina to avoid any cardiovascular risk from changing your lipid metabolism. So, we can alter our basal metabolic rate, metabolic rate. We can change the increased number and size of the mitochondria. And this is done by increasing carbohydrate metabolism, by altering glycogenesis and glucose uptake in muscle and fat cells, by altering the potentiation of the effects of insulin, potentiation of the effects of catecholamines. Remember, we changed our beta-adrenergic receptors, or transcription was altered. We can increase glucose absorption in the GI tract, which will impact, we'll talk about later, which will impact insulin effects. We will drive increase in the synthesis and mobilization and degradation of lipids, which I just talked about, and the impact that has on heart function and your cardiovascular. Thyroid hormones will increase protein synthesis in general. And due to this increased basal metabolic rate, oxygen consumption is also increased in almost every organ, okay? Exceptions are the thyroid, interior pituitary, brain, uterus, testes, and spleen. But certainly in, say, the skeletal system or an adipose system, you'll see a change. Raising this metabolic rate may increase nearly as much as 100% excess thyroid hormones, or decrease by 50% in the case of a deficiency. So with an increase of thyroid hormones, you're going to see this basal metabolic rate go up dramatically, and with a decrease or a deficiency, you will see a decrease. Now, what are some of the specific cardiovascular effects of thyroid hormones? Thyroid hormones have positive ionotropic and chronotropic effects on the heart. So specific actions are via the beta-1 receptor, which can include increased cardiac output by increasing the heart rate. It will alter conduction velocity. It will alter stroke volume, and in general by enhancing contractility. And it will increase renin secretion from the kidneys. Okay? So, cardiovascular effects. Increased cardiac muscle contractility. Again, remember we mentioned that myosin heavy chain gene transcription. This is a key area where the heart is impacted by. And we will increase slow calcium channels. Remember, that was another gene that was altered, which will improve recycling of calcium within the heart. And then finally, obviously, the major receptors for cardiovascular function are these beta-adrenergic receptors, which again will also be impacted. So, thyroid hormones have dramatic effect on our metabolism and specifically also on our ability of our cardiovascular system to maintain. Again, just a reminder, a sort of an overview of all the different ways that this happens. So we've got our circulating T4 converted to T3, binding to our, in majority, dimer with the RXR and our thyroid hormone receptor. This binds to DNA and drives transcription. It drives transcription of metabolic proteins, receptor, beta-adrenergic receptors, channels, myosin. This has an impact on the channels, all, and the respiratory enzymes and all these other proteins on our basal metabolic rate. It's going to alter our oxygen consumption, okay? It's going to have an impact on our cardiovascular output. It's going to have an input on our food intake, and it's going to have an input on our muscle mass and adipose tissue, and eventually on our thermogenesis, which is our temperature, right? So these are our intracellular effects that we talked about, all via gene transcription and translation. And these are our physiology, whole body effects. So this is a good slide for you for review in terms of studying what effects thyroid hormones have on us. So just to go on the review of the metabolic effects in the link, we have basal metabolic rate changes. We have an increase in the number and size of mitochondria, increased carbohydrate metabolism, increased glyconeogenesis and gluconeogenesis, generating free glucose, increased glucose uptake by muscle cells and fat cells, potentiation of the effects of insulin, as we talked about, potentiation of those catecholamines via beta-adrenergic receptor change, increase in glucose absorption by the GI tract that we talked about, and then change specifically around degradation of lipids is a key one, right? And increased protein synthesis. All of these are due to increased in this basal metabolic rate. So those are the physiological effects of thyroid hormone, and we will talk a little bit more about, and just to remind you that the difference between excess thyroid or deficiency can have a swing of double the effect on your BMR. So those are the effects of thyroid hormones and thyroid hormones, how they're made and regulated. And then I'll give you a bit more review before our next lectures, and then tomorrow, and then we can start talking about diseases and what happens when we have these excess or deficiency of thyroid hormone. Okay, well, that's all I have for you today, and